Hello everyone and welcome to our module on cells of the nervous system. There are 5 cell types in the nervous system that you should be aware of, and the first cell type is the neurons, and these are obviously the most important. They are the ones that send out electrical signals to tell our body to conduct actions. Other cells that support the neurons are astrocytes, microglia, oligodendroglia, and Schwann cells, and we'll talk about all of these in this module. Let's talk about what we mean when we say glial cells. Glial cells are all the cells that support the neurons, and I think of them as all the non-neuron cells in the central nervous system. There are some that are called macroglia. These are larger. These are cells like astrocytes, oligodendrocytes, and the ependyma. And then there are cells that are called microglia. When we say that there is gliosis, what we mean is there is proliferation and hypertrophy of glial cells. This is a reaction to central nervous system injury. So these non-neuron cells will undergo proliferation and hypertrophy in response to injury, and astrocytes are some of the most important players. They react significantly to central nervous system injury. We'll talk more about that later. And finally, when we say a tumor is a glioma, we mean it's a tumor of one of the glial cell types. So there are astrocytomas, oligodendrogliomas, and ependymomas, and these are all tumors of glial cells. So in this slide we have a drawing of a neuron, and let's talk about the parts of a neuron that you should know. So up here at the top is something called the cell body, which contains the nucleus, and it gives off little extensions called dendrites. There's then a long portion called the axon, and then at the end there's something called the axon terminal. You'll notice that this long portion has these myelin sheaths around it with little spaces between them called the nodes of Ranvier. We'll talk about that in a little bit in a few more slides. And then there is a Schwann cell surrounding the myelin sheath, and the Schwann cells are what create the myelin in the peripheral nervous system. So this is a peripheral nervous system cell. In the central nervous system, the oligodendrocytes are what create the myelin. So let's talk about neuron action potentials, and to do that I'm going to draw a crude neuron here with an axon and then a little synapse at the end. So when a neuron is sitting at rest, it has a voltage of negative 70 millivolts across the membrane. And what do we mean by that? What we mean is that outside the cell there are positive and negative charges, and inside the cell there are positive and negative charges. But the distribution of these is unequal, so that there's more negative charge inside than outside the cell. And this unequal distribution is maintained by a very high concentration of sodium outside the cell. And then there are potassium ions inside the cell that constantly leak out of the neuron. And this leak out of the cell is what keeps it at minus 70 millivolts at rest. When this cell decides to depolarize, it will open sodium channels and it will allow sodium to rush into the cell. Because sodium is positively charged, it will raise the voltage across the membrane from minus 70 to some higher value. As the voltage gets raised, more sodium channels will open all the way down the axon, and you will have sodium rushing in down the length of this axon. That will be raising the voltage across the cell all the way down the neuron. When the positive charge gets to the end of the cell, it will open calcium channels, and calcium will rush in at the end of the neuron, and then calcium triggers the exocytosis of neurotransmitters. So neurotransmitters are chemical compounds stored in little circular vesicles, and they get spit out of the neuron when this calcium rushes in at the end of depolarization, and that process is called exocytosis. So this whole process is the way the neuron goes from its resting state of minus 70 millivolts to conduction all the way down the length of the axon and the exocytosis of neurotransmitters at the end of the neuron. And the neurotransmitter is what goes and creates the physiologic effect that the neuron wants to generate. If we plot the change in voltage of the neuron over time, it looks like this. So it starts at minus 70 right here. It then decides to depolarize. If the impulses to depolarize are very small, it'll create what's called failed initiations. But if it can reach the threshold voltage of about minus 55, it will then fully depolarize the neuron going up like this. And this rising in voltage here is all created by sodium, and it's very high yield, to remember that. Once as much sodium is rushed in as can, the sodium channels will close, and then potassium will begin to leave the cell, and that will bring the voltage back down. And it's also very high yield to remember that potassium is what brings the voltage back down. It will go through a brief refractory period where the neuron cannot depolarize, and then it will return to the resting state. And this is all very, very quick. You can see the whole process here just takes a few fractions of a second. The clinical relevance of this is that agents that block sodium channel will inhibit the depolarization of neurons, and that's how local anesthetics work. Drugs like lidocaine, benzocaine, tetracaine, and cocaine, these all block sodium channels, and they make it harder for neurons to depolarize. There are also some neurotoxins found in nature that block sodium channels. So the pufferfish is a Japanese delicacy, and the organs of the pufferfish have this toxin called tetrodotoxin, concentrated in the organs. You have to remove those when you serve the fish to people in a restaurant. If it's not removed properly, patients can get very sick. They get dizzy and weak, and they can even become paralyzed. It's a very severe problem. Luckily, it's very rare. Patients have to be supported till the toxin washes out, and you have to flush their stomach with charcoal to try and get any further toxin out of their body. But this just points to the clinical relevance of sodium channels in that when you block them, you can make people paralyzed. Now let's talk about astrocytes. These are very important cells for supporting neurons. They're found in the central nervous system in the gray and white matter. They have a lot of cleanup duties. They pick up extra neurotransmitters. They're responsible for repair and scar formation. And they're a major part of reactive gliosis that occurs in response to injury. So they will hypertrophy and undergo hyperplasia when the central nervous system gets injured. And a very important marker of astrocytes is something called GFAP. This stands for glial fibrillary acid protein. And when you hear that cells stain positive for GFAP or tumors stain positive for GFAP, they're telling you that astrocytes are involved. For any of the cell types you learn, you should always know what the clinical relevance is for your patients. So the clinical relevance of astrocytes is that they cause astrocytomas. These are tumors in the cerebellum of children, and importantly, they will be GFAP positive, which should make sense to you because all astrocytes are GFAP positive. Another point of clinical relevance is that the JC virus infects astrocytes and oligodendrocytes in patients with HIV and AIDS. The JC virus causes something called PML, which is progressive multifocal leukoencephalopathy. It's basically a severe demyelinating disease where HIV patients get encephalopathy, and you have to have a very low CD4 count, usually less than 200 to get this. But astrocytes are a key place where the JC virus infects. Next, let's talk about the microglia. These are the central nervous system macrophages, and they proliferate in response to injury. They differentiate into bigger phagocytes that can go and clean up injured areas of the brain. A couple important things to know about this. HIV can persist in the brain by hiding inside a microglia. And patients who get something called chronic HIV encephalitis, they will have nodules of activated microglia on pathology. Next, let's talk about the oligodendroglia, also called oligodendrocytes. These are cells that myelinate central nervous system axons. Each one of these cells myelinates multiple axons, and that's in contrast to the Schwann cells, which we'll talk about in the next slide. These are the most common glial cells found in white matter, and that should make sense to you. White matter is the myelinated portion of the central nervous system, so it makes sense that you would find lots of oligodendroglia there. And in addition, this cell type is damaged in multiple sclerosis. I talk about multiple sclerosis in another module, but it's a very important demyelinating disorder. So obviously in a demyelinating disorder, you would have loss of the cells that myelinate axons. So the Schwann cells are what myelinates the peripheral nervous system axons, and each cell myelinates one axon in contrast to oligodendroglia. These are very important for neuron regeneration, which we'll talk about in another module, but if you have nerve damage and you're able to regenerate, the Schwann cells are going to be very helpful. These get destroyed in Guillain-Barré, which is a severe demyelinating disease, and these can form something called schwannomas, which are also called acoustic neuromas because they classically affect cranial nerve 8 and interrupt hearing. Let's talk about myelin. This is a very important substance that surrounds the axons of neurons. It's composed of lipids and proteins, and it increases the speed of impulse propagation in the axon. The presence of myelin allows for something called saltatory conduction, and let's talk about what that means. Take a look at this peripheral neuron down here. You'll see that there are myelin sheaths and Schwann cells surrounding the axon with little gaps between them, and those gaps are called the nodes of Ranvier. The presence of myelin allows the depolarization to jump from node to node. In other words, the axon does not need to depolarize its entire length. It can go from node to node hopping along, and this makes the conduction velocity much faster, and it makes the efficiency of the neuron much greater. There are two metrics you should know about that people use to describe how efficient a neuron is. One is called the conduction velocity, and that's easy to visualize. It's simply the speed at which the impulse moves down the neuron. And conduction velocity is obviously improved by having myelin sheaths present. The other one is a little more complex to understand. It's called the length constant. If you measure conduction through a neuron, you will see that it decays over time. So the strength of the signal gets weaker as the signal goes down the neuron. The length constant is how far you can go along the neuron till you've decayed to about 1/3 of your original depolarization potential. So it's a metric of length, and the more myelinated and efficient your neuron is, the longer your length constant will be. And you're often asked about conduction velocity and length constant in respect to multiple sclerosis. In multiple sclerosis, your conduction velocity goes down and your length constant goes down. And both of those are a function of losing myelin. Also, don't forget that in the central nervous system, it's the oligodendrocytes that create myelin, and in the peripheral nervous system, it's the Schwann cells. We classify nerve fibers by their diameter and by whether they're myelinated or not, and I've drawn them here in this slide from the largest, which are the A alpha, to the smallest, which are the C fibers. The A alpha are large myelinated fibers, 6 to 15 microns in diameter. This is what most of our efferent motor fibers are composed of. These also carry senses like touch and vibration and position. The A delta are a little bit smaller, but they're still myelinated. They're 3 to 5 microns in diameter. These carry some senses like cold and pain. And then the C fibers are unmyelinated, very small fibers, and these carry senses like warmth and pain. And these have some clinical relevance, so the smaller fibers are more easily blocked by local anesthetics. So that's why when you get administered lidocaine at the dentist, it blocks your pain, but you can sometimes still feel touch when the dentist pushes on the inside of your mouth. Those C fibers are more blocked by the local anesthetic than the A alpha. Also, there are clinical neurologic techniques to study conduction through fibers, but they're more able to study the large fibers than the small ones. So some patients have what's called a small fiber neuropathy, which means they have problems with warmth or pain, and it's hard to test these because most of the testing techniques only look at the A alpha fibers. Okay, let's talk about how our sensory nerves sense the world, and there are 4 structures on nerve endings that allow us to sense the world. These are called the free nerve endings, Meissner's corpuscles, Pacinian corpuscles, and Merkel's discs, and we'll talk about all these now. So free nerve endings are mostly found in skin. They sense pain and temperature. There are separate free nerve endings for pain, cold, and warmth with different receptors, and these signals are carried to the brain by C and A delta fibers. Meissner's corpuscles are touch receptors. They're located near the surface of the skin, and they're concentrated in sensitive areas like your fingers and your fingertips. This is sometimes called glabrous or hairless skin areas. They get deformed by pressure, and this leads to nerve stimulation. And these signals are carried by the large myelinated A alpha fibers back to the brain. Pacinian corpuscles sense vibration and pressure, and they're located deeper in the skin and in things like joints and ligaments. There are these egg-shaped structures which have a free nerve ending in the middle and a bunch of tissue surrounding it. They get deformed by pressure, and this leads to nerve stimulation, and these signals are also carried back to the brain by A alpha large myelinated fibers. Merkel's discs respond to pressure and position, and they're found in a lot of places, but especially hair follicles. They send their signals by large myelinated A alpha fibers, and importantly, they show a sustained response to pressure. This is called slowly adapting. It means they give continuous information to the brain. And you can contrast this with Meissner's and Pacinian corpuscles, which are rapidly adapting, and they respond mostly to changes in input from the outside world. And an example for this that's easy to understand are thermoreceptors. They respond mostly to changes in temperature rather than a set temperature. So for example, when you get in a hot bath or get in a cold pool, it's hardest when you first go in because there's a big change in temperature, but then you get used to it. Those are things like Meissner's and Pacinian corpuscles. That's a rapidly adapting mechanism where you're mostly responding to the change. Merkel's discs are different. They respond to sustained input and give continuous information to the brain. The last thing we're going to do is talk about peripheral nerves. So when you go into the operating room or you look at a cadaver in the anatomy lab and you hold one of the nerves, you are holding something called a nerve trunk, which is what I've drawn here. So the outer layer of this nerve trunk here is called the epineurium. And inside the epineurium are a whole bunch of things called fascicles. And each fascicle is a bundle of lots and lots of different nerve fibers. There is a structure that surrounds the fascicle, and this is called the perineurium. And then there is a structure inside the fascicle surrounding each of the individual nerve fibers, and this is called the endoneurium. A couple of clinical relevance points here to make. The endoneurium is what gets inflamed in Guillain-Barré, which should make sense to you because that's a demyelinating disease, so you're going to lose the myelin, which is inside the fascicles around the individual nerves. Another sort of clinical relevance thing to know is that you can try to repair severed nerves by a procedure called neurorrhaphy, and that's where you take either the epineurium or the perineurium and you stitch them together and hope that they will regenerate, and you may sometimes see that in the operating room. And that concludes our module on cells of the nervous system.